A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Chief Operating Officer of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to antibody discovery and development. Our expert speaker, Professor Brandon DeKosky, has many affiliations that you can read about in the speaker bio section of the viewer. In this presentation, he will discuss comprehensive analysis and engineering of human antibody immunity. Professor DeKosky is the 2022 recipient of the Antibody Society's James S. Huston Antibody Science Talent Award. This award is given to recognize and encourage outstanding new early career scientists in the field of antibody engineering and therapeutics. Please note, the webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our speaker.
Great. Thank you so much, Jan, for the kind introduction and for organizing this wonderful event. And it's really a pleasure and an honor to talk with you all about some of the work that we've been doing over the last few years. So as Jan mentioned, we're going to be talking about comprehensive approaches in analysis and engineering of antibodies, a lot of that through the window of very difficult drug targets. So I know this is a kind of advanced antibody focused audience, but I want to just give a quick background on where we're coming from in the world of adaptive immunity and what we're trying to do and understand. So of course, our adaptive immune systems are comprised of two major arms that provide specific molecular recognition of foreign antigens. One, of course, is the B cells that are elicited in response to foreign antigens, ideally, sometimes off-target directed against autoimmune antigens. And those have two major effector populations. One is the memory B cells that are somewhat quiescent and not actively secreting, but essentially ready to be reactivated. And then the plasma cells that are actively secreting. So each of us have around 10 to the 10th B cells. Each of those express a single unique antibody. Similar, the intracellular component of the immune response is the T cell receptors. I will talk a little bit about T cell receptors today, as long as nobody reports me, since I know it's an antibody-focused presentation. But we're excited about TCRs too. And of course, TCRs are going to recognize peptides presented by MHCs. And so you've got the intracellular and the extracellular arms of specific molecular adaptive immunity. And TCRs have a similar structure to B cells, with B cells are going to have a heavy and light chain, and T cells are going to have an alpha beta or gamma delta chains. So we're really interested in understanding natural immune responses and understanding highly protective immune responses. We've seen over and over in recent years how protective antibody and T cell responses provide clues to develop effective interventions. And so understanding effective responses to infectious diseases or cancers, or understanding the specific molecular insults that lead to autoimmunity are going to provide ways to combat these diseases. And shown here are a couple of examples. The one on the left is an antibody called D25 that actually enabled the first co-crystal, the first interaction-based structure of this Fab with the pre-fusion form of the RSV fusion protein. And this actually led to some stabilized designs of the RSV fusion protein that are now in multiple clinical trials that are looking to have a lot of success. This technology also led toward the stabilization technologies for the SARS-CoV-2, COVID-19 vaccines that we've all taken and have provided so much benefit to the world. So in parallel, there's also emerging strategies. So one of these is the HIV fusion peptide shown in red. So you can see here the three different protomers of the HIV fusion protein, the spike envelope protein. And that fusion peptide is shown here in red. And there's antibodies that can actually directly target the fusion peptide. And that's one way that we may be able to obtain effective HIV vaccines. And so that's a more developing story. I'll talk a little bit more about that later in the talk. So I think it's familiar to most people in the audience that, traditionally we've used single-cell RT-PCR methods to discover human antibodies. These have single-cell resolution, and they can get the native pairings, both heavy and light or alpha and beta. And those have a somewhat high cost and low throughput. Even the most recent, like 10x genomics types approaches, you're only looking at a few thousand cells versus those, 10 to the 10th B cells in the body, or, tens of millions of T cells in a single human blood draw. And so you're still only looking at a very small fraction. The traditional next-gen approaches can get throughput that's really high. So you can take both B cells or T cells, however many you want, and sequence the alphas and the betas and the heavies and the lights all together. But the problem is that you lose the pairing between those two specific chains, and that means you can't recreate the original immune receptor. And so these technologies have a low cost and high throughput. And so for a long time, we've been focused on how do you sequence paired immune receptors, and how can we translate those data into tangible functional insights, not just sequence-based, but actually functional information. And so in a series of publications, we developed the first and still only approach to analyze many hundreds of thousands of B-cell receptors or single B and single T cells at the same time. And so the way that works is by emulsifying single cells, and then we purify mRNA from those single cells that has both co-localized heavy and light chains or alpha and beta chains on the beads. And then we can recover the beads back and do an emulsion linkage RT-PCR, and that actually can recover physically linked heavy and light chains for those alpha and beta or heavy and light chains. And then using traditional next generation sequencing approaches, we can actually deconvolute the sequences of those, and I'll get to it in a few slides, but the physically linked nature of these amplicons actually enables cloning into display systems. So we can get both the sequence and then the function of those repertoires. So this is just an image of some of the data from one person, one experiment. In one day, there were 130,000 really highly quality controlled heavy and light chain clusters. And I think at the time there was around 7,000 paired 20,000. There was around 19 or 20,000 paired human antibodies in GenBank. So this was a substantial increase in the number of sequences that had been observed at the time. And we can do split replicates and show that there's about a 98% pairing precision for recovering the correct heavy and light chains together. So if you look at the cost of these approaches, this slide is a few years old, so the costs have probably gone up in terms of single-cell RT-PCR. The newest 10x genomics are probably in the few dollars per cell range still. And then these paired heavy and light chain sequencing approaches in bulk are around 4 cents per cell. And so it gives a really tremendous cost benefit for high throughput sequencing of antibody heavy and light chain genes. But of course, sequence isn't the only thing that we are after. We also really want to understand what is the functional capacity of these types of approaches. So not just the sequence, but also connecting that to function. And so in terms of trying to clone these libraries into immortalized display systems, so we have essentially what we could do is put a restriction enzymes at the center of the linker, and at the outsides of the amplicons. And that actually enables us to clone native heavy and lights into a bidirectional yeast display format. So it's a Fab format, which is a little bit more thermostable and a little bit more native than a lot of single chain FV, although single chain FV is also useful for a lot of different approaches. But we went with a Fab to try to get the most native-like display in yeast that we could, to try to present the broadest selection of those human immune repertoires, or mouse or other animal, as desired. And so there's a flag tag and a CMYC tag to look at expression of heavy and lights. There's a basic and acidic leucine zipper, and glycine linker and things like that. And in the end, you essentially can get the native immune libraries displayed on yeast cells. And again, this has been published, so I don't want to go into too much detail, but I'll just share some of the case studies. In the original study, we used this to look at paired heavy and light chain responses in HIV, chronically infected HIV patients, and Ebola virus vaccinees. And then we also have looked at SARS-CoV-2 immune responses in a number of studies, and Zika virus convalescent immune responses. One of the advantages of this approach is that we can look at cross-reactivity and affinity by doing sorts of different antigen titrations or different related antigens. And you can see that in this Zika virus study, where we also looked at cross-reactivity to two different maturation states of the particle. That the flaviviruses have both an immature and a mature form of the virus that are often co-presented on infectious particles in vivo. And then also looking at an orthogonal virus in the flavivirus family, yellow fever virus. This is really important because flaviviruses have antibody-dependent enhancement. And so if you have non-neutralizing antibodies to flaviviruses, they actually infect macrophages. And so you can essentially traffic virus and enhance infection by having non-neutralizing antibodies. So that's why cross-reactivity is really important, and we can study that in the system. So more recently, in some exciting studies, I'm sorry, this paper is actually in revision at this point, but we've actually been partnering with partners at a pharmaceutical company to look at cloning of plasma cells. So in mice that were immunized using a model antigen, we can actually clone plasma cells from bone marrow and spleen, and actually start screening plasma cells for their function. So that's really exciting, because the plasma cell compartment is not accessible for functional screening using traditional antigen bait approaches, because plasma cells don't express antibody on their cell surface. So by doing this, we've been able to learn a lot about the immune responses in that really important plasma cell population that is responsible for secreting antibody in vivo. And we also did some kind of randomly paired controls that we're interested in understanding what are the native versus non-native antibody responses in those populations. Interestingly enough, so there are some affinity bins that we use those titration-based approaches. Those sequences in red actually had K off below the detection limit. And so low, medium, and high affinity, we were able to gate reasonably well, but I think we can be looking at these data and find ways to gate even better. And so many thanks to our collaborators at Sanofi for their support and helping in this experiment together. And hopefully that paper is, again, under revision, and we'll see it out soon. So in terms of understanding and improving antibody evolution, we are also trying to understand a little bit better what are the collections of mutations that essentially can provide the most beneficial antibody improvement or evolution, both either in vitro or in vivo. And so we first started to look at this with an anti-HIV antibody called VFP16.02. It recognizes the HIV fusion peptide, as I had shown on that intro slide. This one was actually elicited in a mouse model of experimental HIV vaccination, and it was a decent broadly neutralizing antibody, but we wanted to know how many mutations was it away, and what kind of things would you need structurally to guide such a vaccine-elicited antibody to having even better breadth and potency. And so a postdoc in the group, Bharat Madan, has took this problem on, and he added individual mutations all along the heavy and light chain variable regions, and then screened those for low, medium, and high affinity. And then through next generation sequencing and bioinformatic analysis, was able to deconvolute the individual effects of each of those mutations. So interestingly enough, if you look at each of these defined single mutations and their impact on the affinity against HIV envelope trimer, what we found was actually 40% of the analyzed mutations were deleterious, 60% provided little to no change, and just about 1% actually could enhance affinity to the fusion peptide. Interestingly enough, a lot of those were concentrated in the framework 2 of the heavy chain, as well as a few important ones in the framework of the light chain as well. And so we dug into these a little bit more structurally and biophysically. We saw that there were some very important correlations, which was really wonderful to confirm that actually if you can enhance a binding affinity to one HIV trimer, that was also closely associated with breadth to other trimers, at least in the fusion peptide epitope. And that was really exciting, and a lot of that is because of the conserved nature of the way that fusion peptide epitope is. So that was really great to see. There were also some interesting things regarding the recognition of the full trimer context versus just the fusion peptide alone that were very helpful for getting higher affinity and breadth and potency. In particular, the flexibility between heavy and light chains. So those mutations that essentially modified the heavy and light chains a little bit were very helpful for getting breadth that essentially added flexibility to most likely accommodate different trimers and fusion peptides. So building on that, we next set out to essentially repeat this with a human antibody that was a little bit more potent already and see how far we could push it. And so a graduate student in the group, Bailey Bonnick, took this one on, and sorry, this slide is also, this paper is also under review now. So sorry, the text in the bottom right needs to be updated. But yeah, so what we did was essentially a similar approach with single mutation additions and site saturation mutagenesis and functional screening. And we also started to combine mutations together. So using things like DNA shuffling and re-SSM to try to really sample fully all the different possible mutations that could essentially guide this antibody to having a much improved immune response. Just a quick note, I won't go into the whole story of the iterative screening approaches, but the first round of screening, we had a really major gap for, there's a number of different very similar fusion peptides that circulate. You can see those in the bottom right of this figure. And the third one down that's shown in orange, that's what we call the, I believe that 1, 2, 3. Yes, that one is 9 amino acids long, and that is the most difficult for these to neutralize because the other fusion peptides are actually 8 amino acids. And so in the initial round, we found that these antibodies were really not neutralizing very well at all against viruses that carried that 1 amino acid insertion. So we started screening with V4, and that's what really led to some transformational improvements. So If you look here, these are flow plots where the y-axis shows antibody expression and the x-axis shows trimer binding. In blue is the wild-type antibody, and then in red is the library. And you can see in the center columns that the single mutants, we saw some pretty good improvement against a number of antigens. A lot of those were more focused on the heavy chain than in the light chain. We're not totally sure why, but it was interesting to see that a lot of these mutations were focused on the heavy chain, where the improvement was a lot more significant. And then in that second round of screening, in the right column, and in particular, I'll draw your attention to the bottom two plots in the right column. You can see that the wild-type antibody shows no recognition against this V4, even at a higher concentration, 7 nanomolar versus 0.3 nanomolar of the very top column. But then after screening the library for a couple of rounds, after shuffling mutations and things like that, we were able to essentially have gain of function against this V4 sequence. And so working with collaborators at Vaccine Research Center led by Peter Kwong and a number of other folks there at VRC, there was a 20 virus panel that was assembled to model the 208 strain panel on the y-axis. And this gave pretty good predictive capacity. You can see that the best antibodies that target the fusion peptide specifically are around 60%-ish broad. PGT151 recognizes an epitope that includes the fusion peptide, but it's not really a fusion peptide binder, although it is on this plot to use because it does recognize a somewhat similar epitope. And so we use that 20 virus panel, and it turns out that one of the variants that we identified actually neutralized all 20 viruses on that panel, although one was somewhat weak in terms of microgram per milliliter, the one on the far right. So this is either 19 out of 20 or 20 out of 20. But when we took that to the 208 virus panel, actually, that ended up being, you can see it in the top right of that plot on the left, in red, VRC 34.01 multi-mutation 28. And that ends up neutralizing about 80% of total strains. So we've essentially expanded this particular clone. It neutralizes a little bit, probably about half or 60% of those resistant strains it now picks up. And it is the broadest anti-HIV-1 fusion peptide antibody reported so far. And you can see that also in the neutralization dendrograms. So each of those kind of different areas on the expanded dendrogram plot are a different HIV clade. And you can see that this new variant picks up a lot of different variants from many different clades around the world. So there were actually 3 structural features that we found that really enhanced binding significantly for this antibody. The first one was an E2K mutation that's actually right at the beginning of the framework 1 of the heavy chain. So just the second residue in the entire 116 amino acids or so of the antibody heavy chain. And this one actually forms some electrostatic interactions with the trimer backbone. It doesn't recognize the fusion peptide directly. It actually recognizes the trimer backbone, and that interaction is somewhat conserved across HIV trimers, so that was very helpful. The second mutation actually did interact directly with the fusion peptide. It's at this 58 residue, changing this threonine to phenylalanine. And that phenylalanine then moves a tyrosine 94 just a little bit, and that allows for enhanced interaction with the G514 residue of the HIV fusion peptide. And then the last one that really expanded the breadth was this A33P mutation, so changing an alanine to a proline, and that actually moves some things around near the binding site and accommodates the binding groove. It makes the binding groove about an angstrom wider. that essentially enables the recognition of fusion peptides of different lengths. So you can see in purple in that structure on the right, that's the fusion peptide inside the binding groove. And that A33P essentially broadens the binding groove to enable accommodation of those insertions. So that was kind of the big mutation that really enabled recognition of a much broader selection. So this antibody is wonderful. It's definitely not there for example, clinical use. But I think what this shows is, if you want to design a vaccine that's going to recognize the fusion peptide and allow for better breadth, this is essentially the way to do it. Getting interactions with the fusion peptide itself, the trimer backbone, and also ensuring that you are able to accommodate those insertions and give a more flexible binding site to accommodate the diversity that's out there. So we've also been starting to apply these antibody improvement approaches to malaria transmission therapeutics. And so, of course, malaria still causes around 400,000 deaths a year. The majority of those are in young children in sub-Saharan Africa who do not yet have established immunity to coexist with seasonal malaria infections. And so there's a big drive for low-cost solutions that would be somewhat low touch points, because a lot of the populations are difficult to treat on a frequent basis. And that's part of the reason that a lot of the small molecules haven't been as effective. And so, if you could identify an antibody that would, for example, one injection, and that would get you through an entire rainy season to prevent protection, that would really go a long way in helping to alleviate the disease burden. And so our colleagues at Vaccine Research Center actually have been making great strides in this in recent years, along with many others around the world. There's a number of different antibodies that have been shown to be potentially beneficial for malaria prevention. In clinical trial, this antibody CIS43LS, it targets what's known as the junctional region of the malaria circumsporozoite protein, and it actually prevents the sporozoites from trafficking from the mosquito bite and infecting in the liver. So in a controlled malaria challenge, they were actually able to show good protection in the clinic. And so we started working with them to improve this, the potency of this antibody, and try and get it to a point where you need essentially less antibodies, so that one injection could give protection for much longer. And so we used a similar approach: VHVL recombination, DNA shuffling, site saturation mutagenesis. And in this case, we weren't totally sure what antigens to screen against. There were a number, and I'll kind of walk you through them on the next slide. But the end assay, because unfortunately there's not really an in vitro assay for this system that is, really strongly predictive, or that can really confirm activity. And so all of these antibodies had to be tested in a mouse challenge, and I'll show some data from that as well. So if you look at the CSP molecule, it's shown here in the center under that bar that says full CSP. You've got the major repeat regions in blue. You've got minor repeats shown in gold. And then there is a junctional region that's shown in the green color. And that junctional sequence is actually one of the two epitopes recognized by this antibody, CIS43, and the other is those sequences in yellow. It does recognize the sequences in blue a little bit. And so since there's only one of those junctional peptides in the CSP molecule, this antibody has very high binding affinity to the junction, and then it has kind of medium or lower binding affinity to the major and minor repeats. And that's maintaining that balance, we have found, is extremely important, because you have to get very high recognition of what we call the junctional epitope peptide 21 that's shown in the top right, And you can see that that peptide has both the green junctional region as well as two major and one minor repeat, versus what we think of as the lower affinity target interaction, which is peptide 25, which is the yellow-blue, yellow-blue. And then what you don't want to recognize, we know now in hindsight, is peptide 29, which is all of these blues. When we started this project, we actually weren't sure how you can get the best recognition. And it's actually in hindsight that we now know from what was protective in the mice what the best antigens are to screen against. But at the time we didn't know, and so we screened against all of these different peptides and a truncated version that cuts out a lot of the repeats, as well as CSP. And so anyhow, cutting a long story short, we added a lot of mutations and did DNA shuffling and screened against all these antigens, and then made antibodies and found out which ones happened to work. And so the one that happened to work the best was this one in the right side of that red box called CIS43 variant 10. It's about 6-fold more potent than CIS43, which was very exciting. So, 6-fold, that's going to get you 2, 4, maybe 2.5 half-lives. And so if you're looking at a half-life of maybe 7 weeks, you're talking about an extra 3 months of protection from a single dose. So that's really exciting. We're continuing in our engineering of this antibody, and you can see in this crystal structure some of the mutations that there's that phenylalanine 27, and then this S85 down in the bottom right, and then I believe that V2 to R2 in the heavy chain as well. And so this is published. You can read about it, but I think one of the most interesting things about this story is how it not just improves this antibody, but tells us what the different balances of affinity against this very strange disordered CSP molecule kind of need to be in order to get good protection against malaria. So the last story I want to tell today, if you'll grant me the opportunity to talk about something very similar to antibodies, and that's T-cell receptors. We're very interested in understanding functional immunity of T-cell receptors against all the different targets. T-cell receptors have a lot of similarities in terms of their recognition and in the structure. And so we've actually found that we can use a similar strategy where we clone capture the natively paired alphas and betas and clone them into displaced systems. And so Ahmed Fahad and Andrew Chung in the group, Ahmed, former graduate student, current postdoc, and Andrew, postdoc, former postdoc in the group, both work together to really push this through. And so essentially the way this work was published just a few months back in Protein Engineering Design Selection. And so we can capture the natively paired alphas and betas from human blood samples and then clone them. In this case, we have to use mammalian display because TCRs do not really display effectively in yeast unless you make a number of compensatory mutations for each TCR. And so we went straight to Jurkat cells. And then you can screen based on affinity, or more recently, we're doing a lot of activation-based screening to understand the different peptide MHC and cellular targets. And one of the reasons that we're really excited about these immortalization technologies for TCRs is that analysis of TCRs from primary cells is extremely difficult because the primary cells themselves have a lot of cellular momentum. So some T cells are exhausted, some are effector memory, stem cell memory, naive, and each one is going to react in a different way based on its cell state. Furthermore, different TCRs have different, affinity versus activation properties, and so it's kind of that nexus of cell state plus, TCR biophysics that leads to potentially productive versus not so productive TCR based protection. And so by capturing the TCR genes, putting them in display libraries, we can screen not just for all those functional features, but also for the many different peptide MHCs that are available in each donor. You've got 12 different HLA genes or MHC genes. Each of those combines hundreds of different peptides. And so there's really just too many to try to screen in primary T cells, even if you could screen primary T cells effectively, but you can't because of that cellular momentum. Anyhow, so that's all the reasons that we're excited about capturing TCR libraries, cloning them into different display systems, and doing different types of screens to understand their function. So this work is published, so I don't want to go into too much detail on it, but you know, we see fantastic enrichment for infectious disease peptides, in this case Epstein-Barr virus-derived peptide MHCs, and we can sequence them and understand and look at affinity sorts and screens and things like that. We can also put them into native human CD8 T cells, and that allows us to do a lot of very important cell activation-based screening on in native primary T cells. So these are things like IFN gamma secretion, proliferation, and other changes in cell markers that you really can't do at all in non-immortalized lines. And you know, so one of the things that we're really looking at is sorting for directly for cell-killing T cell receptors. And so we have actually started doing that in humanized mouse models of cancer, where we'll take T cell libraries and do cell-based co-culture, and we start to look and see. Of course, there's always some T cell cross-reactivity, which is challenging, but we work around it. And so if you have heterologous T cells alone, there will be some baseline level of activation. But then if you take a TCR library in co-culture, and you can see that there's essentially twice the number of cells that are reactive. And so we can sort these cells, take a look and see which are the TCRs that are actually being reactive and targeting that tumor cell. And so with that, just a quick summary. So we're taking native antibody and TCR sequences and putting them into high fidelity display. So we're screening native immune responses as well as engineering antibodies for better recognition of particularly challenging targets where the pathway to an effective antibody is not necessarily clear. And we're also looking at patient-specific models and things like anti-cancer TCR discovery. So a lot of people to thank. The earlier work in paired heavy and light sequencing was done at UT Austin with Andy Ellington, Greg Ippolito, Bo Wong, John McDaniel, and a number of others there at UT Austin. At Vaccine Research Center, my postdoc work was done with John Mascola, as well as Peter Kwong, Barney Graham, Jenny Duick, Nicole Doria-Rose, Bob Cedar, John Missazi, and a lot of other folks who have also stayed collaborators on a lot of the work. And my lab at University of Kansas and MIT, we shared a lot of work today from Andrew Chung, Ahmed Fahad, Bailey Bonac, Xiaoli Pan, Bharat Madan, Matias Gutierrez, and Mateus Oliveira de Souza. And also many thanks to our collaborators I mentioned at VRC, also Ted Pearson at NIAID, Hank Balfour and David Price on the T-cell work, as well as Tim Whitehead at Boulder, and our collaborators at Sanofi, Melody Shasavarian and Emmanuel Vigna. And so with that, thank you all for your attention. Really looking forward to questions. And again, just a pleasure to share our work with you today.
Thank you very much. We are going to have a little chance here for the audience to think about some questions. So we do have one in the box so far. First, great talk, thank you, which I second. The actual question here is, how are you sure about the specificity of B/T cells at single cell emulsification? Or do you sort a bunch of cells and determine specificity later?
Yeah, great question. So I think what, and just to make sure, when you talk about specificity, I think you're referring to functional specificity in terms of knowing what targets they recognize. And so the throughput of these emulsification methods is high enough that we can take pretty much all cells that we have, particularly for B cells. With T cells, we probably can only use about half of what's in a blood draw. We top out at about 5 million or so. But for B cells, there's usually only a couple million in a blood draw. And so we can emulsify all of those, immortalize the repertoire, and then just screen from that stage. And so normally we determine specificity later after the library has been cloned. And the reason that's really advantageous for a lot of our purposes is that we're really interested in things like cross-reactivity to multiple antigens. So if you take HIV or flaviviruses, for example, you want to know what recognizes this broad collection of HIV antigens that is very diverse. And it's hard to pick 3 or 5 antigens that you could even sort with from in the first place. If you're looking at plasma cells, there's no way to sort them for specificity because they don't have B cell receptors on their surface. And so there are some tricks. So you can do like nanopen capture, like with Berkeley Lights, and do kind of like an on-chip bead-based ELISA and then elute the cells that are specific. But you can really only do that for one antigen, and so you can't look at cross-reactivity, for example. So that's really advantageous there. I think with T-cell receptors, you have a similar need to collect the repertoire in an unbiased way, again, because of those dozen MHC genes per donor, plus you have all of the cross-reactivity of different peptide MHCs. I didn't get into it, but the MHC has a lot of similarity and then similar peptides, and so you get cross-reactivity across different MHC genes and different peptides that are related. So yeah, just generally for a lot of our experiments, it's very advantageous to capture the whole repertoire first and then screen for function from that point.
Okay, thanks very much. So just as wait a minute, we do have one more question. I have a follow-up if we have a chance here. But the next question that came in was, due to CDR plasticity, sometimes antibodies show off-target binding after full development of antibody. How can we work on optimizing CDR plasticity, or work to optimize CDR plasticity?
Yeah, I think that's a great question. A lot of the time in an affinity maturation campaign in vitro, you might end up with some solutions that are highly specific, but then also you can end up having some solutions that just provide, kind of sticky binding. So for example, maybe in a phage display library, and you're screening against, a antigen in the immunotubes, and there's some sticky solution. Maybe there's a lot of phenylalanines in the binding site or something. So there's a few ways. There is some good prediction methods that can be used to maybe remove those sequences that are of high risk. There's also a lot of good kind of off-target bait strategies. We actually end up with some of these off-target solutions, particularly when the specific antibodies are very low frequency, so you end up with these sticky ones instead. And having like a non-specific bait's really helpful. So we'll put, a non-specific bait antigen on a different fluorescent color, for example, with yeast, when we can do flow cytometry. And the last is there's some polyspecificity reagents that have been developed that are, I think, really helpful. They're not a defined, some are defined proteins like heat shock protein. Others are actually just like, LC purified biatenylated protein prep of just, a bunch of random proteins. And that's, kind of purified in a highly consistent way. And those are actually really helpful for experimentally removing those problematic off-target binders. So, yeah.
Thanks very much. No technical questions at the moment. If I could just ask one non-technical questions. So you worked a bit with NIAID around the anti-malaria antibodies, and just, again, not to put you on the spot, but to put you on the spot here, do you know what their general plans might be if they did have a successful antibody, in the sense of having successful clinical studies, and they actually wanted to go ahead and distribute this kind of thing? Do they actually have a program in place for a successful antibody for malaria?
That's a great question. I'm not the best person to answer it, but I can tell you, certainly I wouldn't want to share anything that's non-public, but I can say that it's definitely public that Vaccine Research Center does have their own phase 1 pilot plant on site, so they can generate enough antibody to do phase 1 in-house like they had done. And they also have a lot of partner sites in different field trials around the world. And that's where they've done a lot of field trials for vaccines and antibodies, and they have those ongoing. So I won't necessarily say what their plans are, but certainly they have a number of potent antibodies, both from this and from other groups. And they have, again, the pilot plant and the clinical sites. So certainly they could if they wanted to, and I think they want to. So.
Excellent. Well, that's great to hear. And I'll go look for more publicly available information from them on that. Thank you. Mm-hmm. One more quick question here. Have you considered employing mammalian display for your B-cell library display work, similar to your TCR work? Why or why not?
Yeah, great question. That is something that we are doing and are enabling in different ways. I didn't talk about it today. I have talked about it in some other forums and venues. But yeah, that is also something that we're interested in doing. Yeast can give you affinity-based information, but we're very interested in things like neutralization, which you can't really screen using yeast display. So with that'll have to be the subject of another talk. So please come back, next year or something, and we'll be delighted to share more about that.
Okay, well, thank you very much. I think we have exhausted the questions for the moment. As I'm giving my concluding remarks, I'll keep an eye over there. But assuming we are in our concluding remarks, I would like to say, in concluding, I would like to thank Professor DeKosky for providing his insights on the comprehensive analysis and engineering of human antibody immunity. And I thank you very much for joining the webinar today. An on-demand version will be available within a day, and I'll send a link by email to everyone who registered. Please feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.